Hello everyone, my name is Rupsa Parida. Today I am here to present my topic. The name of my topic is ROS act as second messenger for apoptosis. Second messengers are molecules that relay signals from receptors on the cell surface to target molecules inside the cell. ROS means reactive oxygen species, which are highly reactive chemicals formed from oxygen, and it has classified as radicals and non-radicals. Radicals are superoxide, hydroxyl radical, nitric oxide, and non-radicals are hydrogen peroxide, peroxynitrides. Reactive oxygen species have two primary functions. First one is second messenger, second one is pathologic mediators. Reactive oxygen species originate from some environment factors like smoke, smog, UV rays, other carcinogenic compounds, and from some cellular sources like oxidoreductase enzyme from mitochondria, cyclogenase and lipoxygenase enzyme from endomembrane system, and NADPH oxidase. NADPH oxidase is otherwise called as NOx. NOx is included in a redox signaling pathways. The first messenger for redox signaling is uh, vasodilative factors, cytokines, growth factors, and NAD, NADPH oxidase or NOx region is the catalytic site which converts diatomic oxygen into superoxide ion and from superoxide we go through and from superoxide we go through series of events to form hydrogen peroxide. Hydrogen peroxide is then enters into the cell through plasma membrane and breaks down into water and superoxide, and then superoxide is then damages the cell or damages the plasma membrane or damages the DNA. Eventually the cell undergoes apoptosis. So let's discuss the ROS formation in mitochondria. During electron transport chain, reactive oxygen species generate in mitochondria. The inner mitochondrial membrane, nicotinamide nucleotide transhydrogenase enzyme, complex 1, complex 2, complex 3, complex 4, and coenzyme Q is present. During the electron transport chain, there is the flow of electrons in the inner mitochondrial membrane through complexes. In between complex 1 and complex 3, there exists a Q cycle which receives the electrons and passes the electrons from complex 1 to complex 3. During this Q cycle, we get the reactive oxygen species when the rate of electron entry into the respiratory chain are mismatched. Superoxide production increases between complex 1 and complex 4. Partially reduced ubiquinone radical donates an electron to oxygen to make it a superoxide. Superoxide further forms hydroxyl free radical through Fenton's reaction. On the other hand, superoxide dismutase or SOD acts on superoxide ion to form hydrogen peroxide. Reduced glutathione or GSH donates electron for the reduction of hydrogen peroxide by the action of glutathione peroxidase enzyme, and in the reaction we get oxidized glutathione or GSSG. Now cell needs to reduce GSSG to GSH by NADPH, which donates electron and reduce GSSG to GSH in presence of glutathione reductase enzyme, and we get oxidized NADP plus, and again NADP plus reduced to NADPH by nicotinamide nucleotide transhydrogenase enzyme. The GSH oxidized into GSSG by protein thiol reductase, and in that it reduces oxidized cis residue of proteins and enzymes that act as defensive mechanism against oxidative stress. What is oxidative stress? It is a disturbance in the balance between the production of reactive oxygen species, and antioxidant defenses. In this way, we have discussed about the reactive oxygen species and how it acts as a secondary messenger. So let's see in my another slide how reactive oxygen species act as secondary messenger for apoptosis. So let's discuss. Mm, Calcium is an important second messenger, regulates a variety of cellular function including contraction, secretion, metabolism, gene expression, cell survival and cell death. Calcium enter into the cell through transmembrane proteins called calcium channels. Calcium ion communicates with a number of other systems and pathways, among them also with reactive oxygen species such as superoxide anion, hydrogen peroxide, and hydroxyl radicals. Mitochondrial dysfunction causing excessive reactive oxygen species production. Under normal condition, up to 1% of the electron flowing to molecular oxygen through the electron transport chain may be diverted to form superoxide. Endoplasmic reticulum is a major site of calcium storage. Calcium from endoplasmic reticulum is flowing mainly through the calcium release channels as IP3 receptors. IP3 means inositol triphosphate. These channels are accumulated in mitochondrial associated membranes or MAMs, which acts which associates with the outer mitochondrial membrane. Calcium ions from the cytoplasm enter the mitochondria through voltage dependent anion channels VDAC. High level of calcium stimulate respiratory chain activity leading to higher amounts of reactive oxygen species production. Reactive oxygen species can further target endoplasmic reticulum based calcium channels leading to increased release of calcium ion and further increase of reactive oxygen species levels. Increased reactive oxygen species and calcium ions load can open the mitochondrial permeability transition pore MPTP, resulting in the release of pro-apoptotic factors. Reactive oxygen species and calcium can induce endoplasmic reticulum stress in the course of oxidative protein folding in the endoplasmic reticulum. Reactive oxygen species are generated during electron transfer between protein disulfide isomerase or PDI and endoplasmic reticulum oxidoreductin 1 ERO1 alpha. Protein disulfide isomerase can also associate with reactive oxygen species generating NADPH oxidases or NOx, further increasing reactive oxygen species levels in the endoplasmic reticulum. NOx derived from reactive oxygen species can modulate sarcoendoplasmic reticulum calcium ion ATPase activity, thus increasing calcium levels, contribute to endoplasmic reticulum stress, and activate unfolded protein response UPR. Reactive oxygen species can increase calcium ion release to the mitochondria, which activates reactive oxygen species generation in the mitochondrial respiratory chain. Mitochondrial reactive oxygen species can affect NADPH oxidase or NOx, thus further increasing reactive oxygen species and calcium load in the endoplasmic reticulum in a vicious circle, ultimately leading to apoptosis. In this way, I have explained my topic. Thank you.